away. Can you hear me, Professor Langer?
I hear you fine, yes.
Okay, great. So greetings to all attendees joining us on Zoom and YouTube from across the world. In fact, going by the registrations, we had people from all parts of India, North America, Europe, Africa, Middle East, East Asia, Australia. It's an honor and privilege and delight for Venture Center in Pune, India to host today's talk by Professor Bob Langer. My name is Premnath, and I'm the founder director of Venture Center, located in the CSIR National Chemical Laboratory campus in Pune, India. The Venture Center has just finished 15 years of service, growing from humble beginnings in 2007 to India's leading inventive enterprises incubator today. In more ways than one, the Venture Center initiative is inspired by and a product of what MIT and Professor Langer champion, namely science-led innovation, real-world, transformative and impactful problem solving and inventive enterprises. I have been a keen follower of Professor Langer's work since the early 1990s, and he's always been an inspiration for me personally and the Venture Center's extended network. Today's talk is being organized on the occasion of Venture Center finishing 15 years. It's also part of the Innovation and Technology Enterprise lecture series that aims to present exemplary cases of research-based innovation translated into technology and business ventures, the associated ideas, the people, the experiences, in order to inspire, energize, and inform scientists, technologists, and business people into action. I cannot think of anybody better than Professor Langer in the world today, who embodies that spirit and is and he's clearly the right person to inform us and inspire us on this topic. Professor Robert Langer is one of the 12 institute professors at MIT. Being an institute professor is the highest honor that can be awarded to a faculty member. He has written over 1,500 articles, which has been cited over 360,000 times. His H index is 298, is the highest of any engineer in history, and fourth highest of any individual in any field. His patents have been licensed or sub-licensed to over 400 companies, and he's a co-founder of a number of companies, including Moderna, which is very much in the news currently. So Dr. Langer served as chairman of the FDA's Science Board, its highest advisory board, from 1999 to 2002. His over 220 awards include both the United States National Medal of Science and the United States National Medal of Technology and Innovation. And he's one of the 3 living individuals who have received both these honors, the Charles Stark Draper Prize, often called the Engineering Nobel Prize, the Queen Elizabeth Prize for Engineering, which is again the top prize in engineering, Albany Medical Center Prize, Breakthrough Prize in Life Sciences, Kyoto Prize, Wolf Prize for Chemistry, Millennium Technology Prize, Priestley Medal, the highest award of the American Chemical Society, the Gairdner Prize, Hoover Medal, and the Dreyfus Prize in Chemical Sciences. He holds 36 honorary doctorates and has been elected to the National Academy of Medicine, of Engineering, and of Sciences, and of Inventors. I think we are really honored to have you give the talk today. Today's talk by Professor Bob Langer is titled From the Lab Bench to Clinic, How to Solve Global Health Challenges by Developing New Therapies Through Innovation and Founding New Companies. With these words, I welcome Professor Langer to begin his talk. And Professor Langer, as mentioned, you can tell me when to change the slides. Yeah.
I will do that. Thank you so much for such a kind introduction. So I thought I'd tell you a little bit about my career and then give you some examples of how we tried to move from the bench to the clinic and how companies enabled us to do that. So I'm a chemical engineer, and when I graduated in 1974, almost all my colleagues went to work in the oil industry, but I wasn't excited about doing that. I was interested in trying to do something to help people in health or education. And actually, I got 20 jobs in the oil industry, but I didn't take them. And then nobody would hire me in the other areas. But one day, one of my colleagues in the lab said I should write to a man named Judah Folkman at Harvard Medical School. And I wrote him, and if we could go to the first slide, he offered me a job. And I was the only engineer in the hospital. And Dr. Folkman's idea is shown on this slide, that it's about how tumors grow. And when you look at the lower left, what you see is the tumor cell becomes abnormal. It grows in a three-dimensional mass. If you go to the middle bottom, it gets to be maybe 1 million cells. And then it runs into a nutrition problem. It can't get larger than that because cells in the center die. They can't get nutrients or get rid of waste. But the tumor, he said, solves that problem by secreting a signal he called TAF, or tumor angiogenesis factor. And that causes surrounding blood vessels, which are normally stagnant, to grow directionally to the tumor. And that solves the nutrition problem. When you go to the upper right, you see the tumor now getting bigger and bigger and bigger. And ultimately, if the top right, it's very vascularized, and then it can spread through those blood vessels. That's a process called metastasis, which eventually kills. Now, when he explained this to me, I thought this was an incredible theory. Turned out almost everybody in the world didn't believe it. I didn't know any biology. I thought it was great. So my job, he said, was to prove he was right, and in so doing, isolate the first blood vessel inhibitor. One of the things that was key to isolating it was creating a bioassay, and there was no way to study blood vessel growth at the time. So we had to invent a bioassay. And our idea is shown on the next slide. That what we might do is take a eye of a rabbit, put a tumor in it, then some drug delivery vehicle like a polymer or lipid, and then deliver it over a period of time. And the blood vessels would come into the eye because of the tumor. I'll show you that later. But when we first talked about trying to create systems to deliver large molecules, which these angiogenesis inhibitors were, almost everybody that we talked to said you couldn't do it. On the next slide, this is what they basically said, that large molecules can't slowly diffuse through solid materials any more than any of us could walk through a brick wall. They're just too big. The literature actually said something similar. Next slide. And what it said was the use of polymer matrices for slow release systems was virtually restricted to small molecules. But if we were going to solve this problem, we really had to develop these delivery systems. I spent 2 years working on this. I tried many different ideas, and I found over 200 ways that this wouldn't work. But eventually, next slide, I found a way to make little microspheres or nanoparticles, which you see here on the top left and cut in half on the bottom. And we published in the journal Nature, next slide, the fact that you could use this to deliver molecules of any size. In fact, this was the first report, to my knowledge, that you could even use materials to deliver nucleic acids, which we also showed in that paper. Nonetheless, when I first presented this work at a scientific meeting, also in 1976, and I was like a young scientist, 27 years old, people ridiculed it. On the next slide is what they basically said. That what they said, these were some very famous scientists, was that, again, the large molecules can't diffuse through solid materials, and that the solvents I was using, which were organic, to do the encapsulation would destroy peptides, proteins, or nucleic acids. And actually, things went downhill from there. After that, I tried to get grants. My first 9 were rejected because people didn't believe this was possible. And then I tried to get academic jobs, teaching jobs. No chemical engineering department in the world would hire me. They all said this bio stuff that I was doing didn't make any sense. So Dr. Folkman knew a man named Nevin Scrimshaw, who was in the nutrition department. And he asked him, well, would he hire me? Now, Dr. Scrimshaw was a great man, but he was kind of like a benevolent dictator. He liked me, so he offered me a job, but he didn't ask anybody else in the department what they thought. That would have probably still been okay, except for one thing, which was the year after I joined the department, he left. So a lot of the senior faculty decided to give me advice. Their advice is I should leave too. In fact, the next slide shows what one of my colleagues was recalling, This is Michael Marletta, who's a very famous chemist now at Berkeley. And he said, describing me in the late 1970s, he said, one evening he went to a faculty dinner at a Chinese restaurant with me and some senior MIT professors. He said, a senior scientist sat quizzing us while smoking a cigar. He said, when the older scientist heard my concepts for drug delivery, he blew a cloud of smoke in my face and said, you better start looking for another job. So that was very discouraging, but I kept trying. And as I mentioned, if we could go to the next slide, one of the things that I wanted to use this for was this bioassay, which we published in Science. And I want to give you a warning before I show the next slide. But what we were looking at was, could we stop blood vessels from growing? We had literally 100 different fractions, and we did probably 2,000 eyes. And the next slide is a little bit gruesome. It's just a representative picture from one of these thousands of studies we did. We also quantitated it in this Science paper. And the next slide just shows a picture of what you see. If you don't have the inhibitor, which is on the left, what you see is after, say, a number of weeks, the blood vessels form this sheet and they go right to the tumor. If you let these animals live, you would see the tumor would eventually become three-dimensional. It would actually kill. We sacrificed the animals before that. But if you put an inhibitor in, and you see that in the right, what you see is the blood vessels are sparser, they're lower, and they actually avoid the tumor region. We published this in 1976, but it took another 28 years and many companies like Genentech doing great work, building on what we did before the first blood vessel inhibitor was approved by FDA. But on the next slide, what you see is kind of remarkable. 28 years later, one after the next blood vessel inhibitor is approved by the FDA. And Avastin, which is the first of these, is probably the #2 best-selling biotech drug in history. And these are used by tens of millions of people every year to treat cancer and also eye diseases like macular degeneration and diabetic retinopathy. Dr. Folkman and I also thought about filing a patent. At that time, Children's Hospital never had a patent. That's maybe one of the best children's hospitals in the world. So we filed a patent, but 5 years in a row, the patent examiner rejected it. He said he didn't really understand it. And the people at the hospital said, Bob, you're wasting so much money, you've got to give up. But I don't like to give up. So I started to think, how could we convince the patent examiner, legally, of course, to allow this patent? And science really wasn't working. But as I told you, when we first did this, everybody said it was impossible, it couldn't work. I wondered, maybe someone wrote that down. So I did what's called a science citation search in 1982. This was 6 years after that Nature paper to see, and after 5 rejections from the patent office, to see if anybody ever said anything about our work. And actually quite a few people did. The paper has been cited well over 1,000 times. But if we could go to the next slide. And I had no idea this was written. This was from 5 of the top material scientists in the world. And they're describing this field. And this was published in 1979. And I really didn't have any idea that they wrote this. But when I did the search, it came up. And they're describing this field. And they said, generally, the agent to be released is a relatively small molecule with a molecular weight no larger than a few hundred. One would not expect that macromolecules could be released by such a technique because of their extremely small permeation rates. However, Folkman and myself have reported some surprising, that's a really good word for a patent examiner, results that clearly demonstrate the opposite. So we showed this to the people at the hospital. They flew down to Washington to show it to the examiner. And he said, gee, I had no idea. I tell you what, I'll allow this patent if Dr. Langer can get written statements, written affidavits from these 5 people that they really wrote this. So as a young professor, I wrote them, and they were all nice enough to write me back they really wrote it. And the examiner allowed the patent. The next slide just shows that. It was a very broad patent on controlled release of any macromolecule. And with that, we would license things to companies, and I got involved in starting companies. One of those companies, next slide, was a little company called Enzotec. And one of the key things was the first 4 people working there were my students. That's actually very important because they were highly motivated. To really take what they had done in the lab and get this out to the world. We did a merger with a company that was right downstairs from us, Alchemies. Today, that company has 25 products, all microspheres or nanospheres, that are FDA approved or in clinical trials. And it's led to new treatments for schizophrenia, alcoholism, opioid addiction, diabetes. They've got about 2,000 employees. And there are many, many products. I'll just show you some pictures on the next slide that are based on this discovery. If we go to the next slide. And many of them are microspheres that can deliver drugs to treat prostate cancer, endometriosis, schizophrenia, prevent pain, treat type 2 diabetes. Drug-eluting stents are based on this. That's on the bottom right. And so these are used by tens, if not hundreds of millions of people all over the world. So that's actually how I got started doing this with patents and companies. But I'll give you a few more examples. A couple of years later, David Edwards, a young scientist, came to see me, and he was mostly a chemical engineer. He was a chemical engineer, but he's mostly doing math. And one of the things that he did a lot of work on before he came to see me was modeling the lung. And since I was interested in drug delivery, I told him, a big challenge is, could we develop aerosols that would work really well? And aerosols, by the way, at that time were incredibly inefficient. If you took an aerosol, you're lucky to get 2% of it from the device to the lung. You might ask, why is that? Well, everybody always thought before we were involved that aerosols had to be incredibly tiny. Otherwise, they would settle in the back of your throat. But the problem is that when they're so tiny, they have huge surface area. Like it's almost like wet sand. So you aerosolize it and it sticks together in the device, in the air, in the mouth. So very little gets to the lung. So if we could go to the next slide, what David thought about when I mentioned this is, before us, everybody always designed aerosols that were non-porous, which could be either water, like nebulizers, or other types of inhalers. Even at that early time, this was 30 years ago, people had developed dry powder inhalers. But all those aerosols always had a density of like water, a density of 1 gram per cc. They were relatively non-porous. So what David thought about is, what if he makes them much bigger, but very porous? And if you did that, we theorized that then they will float, he modeled this, they will not settle in the back of the throat because of their density so different. They'll float into the lung. But because they're so much bigger, they won't stick together. Like wet basketballs aren't going to stick together to the same degree as like wet sand, right? So they'd be much easier to aerosolize and flow. Also, we felt they'd last longer, because what happens is when something's aerosolized, it goes into the lung, and there are macrophages in the lung, and they eat the aerosols. And normally it's like eating a small meal. Now what they see, it looks like eating a big meal, so it's just slower. So we made some of these aerosols. This was done by Justin Haynes and Giovanni Caponetti, Noah Lotan. And what you could do is, this is a scanning electron micrograph. They're incredibly porous. In the United States, I don't know if you have wiffle balls in India, but we have, this is what they look like, a wiffle ball, very, very porous, in contrast to like a golf ball or a baseball, which isn't porous at all. And so this is what they look like. And if we go to the next slide, what you see is that in an in vitro experiment, which is what's called an Anderson impactor, if you go to the right-hand picture, you can get 10 times more deposited porous to non-porous, which was remarkable. And if you go to the next slide, this is now in animals. If you look at different lobes of the lung, again, 10 times more in porous compared to non-porous. And if you do a human experiment, next slide, it actually could last for days because the macrophages don't eat them. So this was all very exciting. We published this in Science, and it really changed the field. We also, again, filed a patent. I won't do this every time, but the next slide shows you a patent here. And it's a very broad patent again. It's a particulate system for drug delivery to the pulmonary system. And our particles generally had a density of 0.1 gram per cc. So we're way lower than what we put. And the particles were much bigger than ever before. And notice the claim is any agent. So you could use this for anything. So we started this company, AIR, Advanced Inhalation Research. And next, with David, next slide. And this just shows you what happened. We did the initial studies in 96, published it in Science in 97, started it again with a number of our students in 98. And Terry McGuire from Polaris, he was an investor, but he also did a lot of the business work. And immediately we were able to get major deals with 3 big pharmaceutical companies, Lilly, Glaxo, and Pfizer. And we started 2 clinical trials in the first year. And David decided he didn't want to do this forever. So about 4 companies wanted to buy AIR. And what would happen is the investors got great. This is, I think, one of the highest, maybe the highest in history, greater than 1,000% IRR. I didn't even know what IRR was before this, but it's your annualized return. And then, we actually, sometimes people say when you do the things like this, it's bad for the developing world. But actually, what happened is we couldn't get any money to do it at all. Normally, we would write grants to the NIH. They all said it was impossible, wouldn't fund us. But because the investors put money in, when we now proved it was not only possible, but it worked, and it worked even in people. And so David wanted to start a nonprofit to help people in the developing world, and with this technology, and he did. And we never could have done this without the company. And then they spun off another company, and that got acquired. And the next slide just shows one example of what's happened, which is really interesting, that you can take this tiny little inhaler, you can put it in your hand, and this is now used for treating patients with Parkinson's disease, for levodopa. The beauty of this little inhaler is you can do 70 milligrams, whereas before this, you could only do about 2 milligrams. And we're actually working with the Gates Foundation to help to use this kind of technology to help little children with various respiratory distress syndromes, where they don't have power or any other kinds of things, so that we could do aerosols like that. The next example I wanted to give you was started by 2 of my Indian students and postdocs, Ram Sesascaran and Ganesh Venakatraman. And if we could go to the next slide, what we did is people before us had started companies based on proteins or nucleic acids, but they're always monodispersed, meaning that all proteins are about, are always going to be the same. Like insulin, all insulins are 6,000 molecular weight. But polysaccharides are much more complicated. Like heparin is polydispersed. There's all kinds of different molecular weights and compositions in heparin, and that makes it polydispersed. And what we decided to do for the first time, next slide, was create a glycomics company on polysaccharides. And what we thought about was shown on the next slide, that you could fractionate any of these mixtures, right? Any of these polysaccharide mixtures like heparin, to find particular glycoforms. Now, since the entire mixture is FDA approved, you'd like to think that the glycoform would be approved because it's just one component. But nobody had ever before, Ram and Ganesh, ever was able to sequence a complex polysaccharide because of the structures and because of the polydispersity. And what they did is develop the very first approach to do it. I won't go into the science too much, but basically what they did is they could take a molecular scissors, which are heparinases and heparanases, actually, which I was able to isolate with some of the team. And then Ram and Ganesh also used mass spectrometry to look at the cut pieces, and then they got the sequences, and then they put those sequences back together by a mathematical model they developed. And again, this is based on 2 papers in Science. And the idea is that you could have these various potential products shown on the next slide. And we started this company called Momenta. Next slide. And this is Ram and Ganesh. We started, the 3 of us were founders. We got, again, funding from Polaris because they made a lot of money from the last company that I just mentioned. We got other venture capital groups coming in. We went public in 2004. Now, one of the ways that I've often used to finance, or the CEOs use to finance these companies, is you give little pieces away of the technology to big companies to hopefully create a win for those companies, but also a win for us. So we did a couple deals with Novartis, Baxter, Mylan, and CSL. And each of them would get a different like polysaccharide that we might help them with. And then we continued to raise money, not only from those companies, but by going public in 2004, and then with these various deals. What was really exciting is we were the very first company to get a low molecular weight biogeneric approved, low molecular weight heparin biogeneric approved. This is Lovenox. And it was the first complex drug ever approved based on just purely analytical data. And it was the largest syringe launch in history. All the stock analysts, by the way, always said that we would never get it approved. And they said like much larger companies like Teva and others would get it approved before us. This is now actually 12 years later, 2022. They still haven't gotten it approved. But at any rate, 2 years ago, or actually less than 2 years ago, Johnson & Johnson acquired Momenta for $6.5 billion. I just wanted to show you some pictures. On the next slide is a picture of the enoxaparin. And of course, on the next slide, what happened is that the company did very well when they got the FDA approval. But I want to go over one other thing that I think is so important about the work that was done here. And that was solving major public health crises that people may not remember, but 14 years ago, there was a huge heparin crisis all over the world. I'll just show you some headlines on the next slide. And these are just a few, but there were many deaths in many, many different countries all over the world. These are just a few early headlines. And I was once in charge of the FDA Science Board, their highest advisory board, and Ram had a lot of involvement with the FDA too. So they called us up and they said, well, could we help? And so what we did is Ram and myself at MIT, Ganesh, who was chief scientific officer at Momenta, and the Momenta team collaborated with the FDA and the CDC. And on the next slide, we published these 3 articles, which ended the crisis. The first bottom one is in Nature Biotech, which identified the contaminant. And the top 2, which again, we published with FDA and CDC, Center for Disease Control, showed what the problems were. And after this, there were no more deaths. I thought I'd give you 2 more examples. And the next one is kind of serendipitous. If we could go to the next slide. People, I'm sure, know that there's a lot of cell therapy techniques being used for different things. And people try to insert things into cells to make CAR T cells or other things by electroporation, cell-penetrating peptides, or nanoparticles. But if you could press the button, I wanted to just, for the next slide, so to speak, I want to show you that there's issues on all of these, like damage or modification or so forth. If you could press the next slide. So there are problems on all of these. So what we did, we wrote a grant to the NIH to, maybe if we stop that just for a second for this video, or we can show it again. But what we did is we decided to do fundamental studies to look at, this was done by Armin Shari, our student, to look at how cells, you could take a gene gun and get something to enter the cell. And Klaus Jensen, my collaborator, he did a lot of microfluidics work. So what we did, and now you can go to the next slide, is that, and this is a video of a cell going through a constricted tube. And on top of that constriction, you see the gene gun. So we kept inserting it, and you could do 1 million cells a minute and see what happens. But one day, Armin decided to just take the gene gun away. And it was amazing. He got exactly the same result. What does that tell you? And what did it tell him? It means you don't need the gene gun. Somehow going through the constriction creates like a nanopore or something that anything can go through, and then it closes. So what we were able to do, we published this in PNAS, and the next slide just shows what you can do. Next slide. You know, you can actually make these chambers, and we published this in PNAS. And just to show you how effective it is, next slide. You can look at the 4 Yamanaka factors. These are the genes that can create what are called iPS cells. That's a stem-like cell. If you take a fibroblast, you insert them, and you can insert them with a cell-penetrating peptide. You get 2 colonies. nucleofection, that's actually the standard. That's like electroporation. You get 11. But with Armin's device, we got 150. And not only is it more effective, it's actually safer. And one way to look at that survival, and cells survive better. But another way is to look at, is to look at the transcriptome, which is shown in the next slide. That's the messenger RNA profile. And if you look at this, look at the control not treated on the left, and you compare that to electroporation, and this was mild electroporation in the middle, you see huge differences. Whereas if you do the squeeze approach, the differences are much less. So it's a much safer approach. And so in 2014, right after we published this, Scientific American came out with their top 10 advances, top 10 world-changing ideas. The next slide just shows what Scientific American wrote. CRISPR-Cas9, they said, was first, and the squeeze approach was second. I felt I had the order wrong, but that's okay. CRISPR's pretty good. And Armin had started after he did this work as a postdoc. But one day he came to see me, and he wasn't that happy. I said, well, what would make you happy? He said, well, starting a company on this and being the CEO. So we did. I introduced him to Amy Schulman, who is at Polaris. And she became the executive chair. You know, we wanted somebody with good business experience to help guide him, but he became the CEO. We got Howard Bernstein, one of my top former students, to be the CSO, Chief Scientific Officer. We started Squeeze. And on the next slide, what they were able to do was get $1 billion immuno-oncology deal with Roche a few years later. That's actually in clinical trials now to make better B cells by using this approach. Company's a public company, and Armin with over 100 people and doing pretty well. So I thought I'd end this talk with one other thing, and that involves some nanotechnology work we did. If we could go to the next slide or video, which has sound, I believe.
He starts with a nanoparticle of anti-cancer drug. That gets encased in a plastic that releases the drug over time. That in turn gets a special wrapping that disguises the package as a water molecule to fool the body's immune system. And last but not least, the address where it should be delivered, a key that will only fit the lock of cancer cells.
So it doesn't really blow up the cell like that, but certainly we started using this for small molecules and for gene therapy, like siRNA and mRNA. And of course, one of the things that I was involved in was helping get a company called Alnylam started as a member of their founding scientific advisory board, and they used a nanoparticle. In fact, one of my other students, Kenna Kink, led a lot of this work to deliver siRNA, and that would lead to Onpattro, which is now FDA approved. But in 2010, going back in time, if I could go to the next slide.
He starts with a...
is Derek Rossi, Ken Chen, Nubar Fayan, and myself decided to start a company to do almost the opposite of siRNA, to do messenger RNA and create therapeutics from that. And this is the central dogma, as you probably all know, that DNA makes RNA makes protein. And of course, by 2010, protein therapeutics had become huge, but it takes a long time to make the proteins. Whereas if you use messenger RNA, you could make it a lot faster. And because all you'd need to do is have the messenger RNA, and if you could protect it, which you can by putting it in a nanoparticle, and inject it into the body will do all the work and make the protein. And as I'll show you, you can actually predict in a day and make the messenger RNA incredibly fast. So we started this company, the four of us, in 2010, called Moderna. And just to show you the profile of that, Nubar was at Flagship, and they invested $1 million. And we hired a terrific CEO, Stefan Mansell, who Nubar knew and I knew somewhat. And we kept raising equity. Now, again, what I mentioned to you earlier we just kept using the same model. We'd give pieces away to large companies in exchange for funding and help. And so in 2013, we got $280 million down payment from AstraZeneca for 40 targets for heart disease, and also $180 million milestones plus royalties. 2014, Alexion, another $100 million. And each time we did this, of course, the value of the company goes up, so we raised equity too. 2015, Merck, we did a deal on infectious diseases. If we could go to the next slide. So we dosed the first patient in 2016. And then again, we've got more deals, Merck for personalized cancer vaccines and Vertex for cystic fibrosis, and again, raised more equity at a higher value. In 2018, we did the largest IPO in history. And now, and I should need to update this, now we actually have 23 different clinical trials going. The COVID vaccine has been approved for emergency use authorization. We've, of course, expanded our team a lot because of manufacturing, and so now we have, this was now almost double that number of employees. And just to show you the Moderna pipeline, next slide. So COVID is used, the mRNA vaccine is used worldwide, but there's also flu vaccines that are in advanced clinical trials, COVID flu vaccines, combinations, RSV vaccines, but there are also, other types of vaccines like from cytomegalovirus, Epstein-Barr. On the next slide to show you a few more things that are in clinical trials or close to it, AIDS vaccines, Zika vaccines, treatments for autoimmune diseases, personalized cancer vaccines, new cancer treatments. The bottom one is actually the AstraZeneca one, which is in phase 2 for myocardial ischemia. Next slide. And now you see that you can do intracellular diseases. That's one of the other huge advantages of messenger RNA. You can not only do things fast, but you can treat diseases inside a cell. And finally, the cystic fibrosis one that I mentioned. Well, one of the things that, of course, everybody knows is, I expect, is that Moderna was the very first company to make a COVID vaccine. And here's the timeline, which is really quite remarkable. Next slide. So you may know that the Chinese scientists published the sequence on January 11th. In less than 2 days, we had it designed. In just a little over a month, we had the vaccine batches made and ready to send to the National Institutes of Health in Washington for testing. And we were able to treat the first patient, just about 2 months later. And as people may know, it was 95% effective in the clinical trials. And if you look at any of the clinical data published in the New England Journal of Medicine or Nature Medicine, the Moderna vaccine is clearly the most effective. Pfizer BioNTech is next most effective, but the mRNA vaccines, I think, probably work better than anything. Nonetheless, when we did this work, just like I was criticized when I did my early science, people all over the world criticized us because they said the science wasn't any good. Our local newspaper, the Boston Globe, next slide. This was their headlines after we did the first clinical trial. A lot of scientists would come up and say, this isn't how you do science. And so this is front page headlines in May of 2020 after patients were treated. And they basically said, this is not how, a lot of people said, this isn't how you do science. Nonetheless, as you all know, this is now, as we go to the next slide, used all over the world. And hopefully this coming year will make over 3 billion doses. So we can get it to many, many places. So I think I'll stop here. I think that these are some examples of what we tried to do. To me, going from the bench to the clinic is really important because I not only want to see us do great science at MIT, but I want to see us do things that will really make a difference worldwide. And it's because of our students and these companies that's been able to happen and over and over again. Thank you so much for having me.
Thank you very much. Thanks a lot. Would you want me to unshare, or there are a couple of slides that you want me to keep retain for now?
Why don't we see what the questions are?
Yes. So, by the way, I have compiled some of the questions we have already received. So, in the meantime, I will request my colleagues to track the questions as they come along on the chat box as well. Professor Langer, let me just start by a set of there are a few themes that came up when we were looking at the questions which were posed online. But first of all, so let me dive straight into it. I know you will be short of time, so I've compiled some. There's a set of questions which have come up from several people in from Pune, Mumbai, Bangalore. The question is, how do you go about selecting scientific problems to work on? Do you have to weigh between scientific merit versus commercial value? Do you encounter any conflicting motivations or difficult choices in these goals? How and when do you decide that a piece of work is worth commercializing? And how do you decide whether you should commercialize using a licensing route to larger companies or a startup route? And of course, a related question, people wondering if small companies and individuals can really make a big difference when it comes to healthcare, where large companies seem to be the major players.
Wow. Let me see if I can remember all those. But let me start at the back end, because I remember those better. So, well, I think the answer to small companies, there's no question large companies can make a big impact. But let's take messenger RNA. No large company would take a chance early on it. There's only, by far, the two companies that have made the biggest impact are Moderna, and we did it all ourselves. And we were a tiny company, and the other company is BioNTech. Pfizer did a deal with them, but if BioNTech hadn't done all the initial work, I mean, Pfizer wouldn't be able to have done anything. So really, I think there's no better example than what happened on these COVID vaccines to show you that small companies can make a giant difference. And, no large company was able to do anything. In fact, they all refused to do anything. The only one that even did a little bit was Pfizer, because they gave a lot of money and helped BioNTech. So I think that's certainly the case. If we could actually go to the slide we left off on, I will try to answer a question that somebody asked: when do I decide to start a company? I'd say it has to have these six features. First is a platform technology, and that's something you can use over and over again for different things. Most of what I talked about, like mRNA, is a platform technology. Nanoparticles is a platform technology, right? Because you could use it for heart disease, for cancer, for COVID, and so forth. So why do you? So I would say that if I was developing a single product, right, just one product, then it might make sense to license it to a large company. But if I did, let's say I just licensed it to Pfizer. I mean, nothing against Pfizer, great company, but they would just use it for their drugs and for their things. Whereas if you keep it and you do deals like Moderna's done with Merck and with AstraZeneca and with Vertex and by ourselves, the world is benefits much more, and so does the company, because you can have many more products with the same technology. It won't just sit on the shelf. So if we have a platform technology generally, and it's advanced enough, which we'll get to the other bullets here, then very often we do start a company. And that sounds, so the next bullet is a product company. The next bullet is that, we're reasonably far along. We have a seminal paper in a top journal like Science or Nature. And the fourth bullet, that we have really good intellectual property, which I was trying to illustrate. And the fifth bullet is the journey is long, particularly in medicine, so that we have some in vivo proof of principle. And finally, that some of the people in the lab really believe in this so much, they want to work there. And they have key roles, like I mentioned, Armand and Howard Bernstein at Squeeze. And this has been true for pretty much every company, at Ganesh and Ram at Momenta, that people really want to take what they did in the lab and get it out to the world. And the reason that's important is, and you could probably see this if you looked at Apple Computer, right? Apple Steve Jobs started it and they did great. And then they said, well, the board said they need a professional CEO because he doesn't have enough experience. So they fired Steve Jobs And for 15 years, they did terrible. They hired one professional CEO after the next. They were a disaster. Then Steve Jobs came back and they did great again, became the most valuable company in the world for a while. And same thing with the students. They may not have as much experience, but they'll walk through walls to make this happen. And you complement them with people who do have experience. So those are when we decide to do those things. I realize I probably didn't answer every question, but hopefully I answered some.
Sure. One of the other questions on how do you go about selecting a scientific problem to work on, also related to that is, when you come up with these ideas, what is the actual process of connecting the dots? Perhaps, I'm interpreting it as connecting problems and solution, pieces of the solution. I think in Moderna too, you connected developments in mRNA and lipid nanoparticles and so on and so forth at some point and brought them together. So how does that process actually work out?
Yeah, so the way I select ideas for the lab is they can come from anything. They can come from me dreaming up something, somebody in the lab dreaming it up. They also sometimes come from companies that have seen what we do and are willing to give us funding to take us a lot further. We have programs, for example, with Novo Nordisk and Roche and so forth, because they've seen some of what we have done and on platforms and want to apply it in certain areas. It has come from foundations, like, again, Bill Gates has come to see me a number of times, and we are working on a lot of developing world problems, that he and his team have brought up to us. So that's how, but that doesn't mean we'll necessarily start a company, though we have started companies in a number of those cases. Connecting the dots, the second part of the question, that's done on a case-by-case basis. You just have to see, well, what do you need to solve the problem? And usually it's going to be multidisciplinary. And we have, fortunately, a lot of very smart people in the lab and with different disciplines, and we work together to connect those dots depending on what the problem is.
Thank you. I'm just restricting myself to questions which mostly are for the younger people and younger scientists and researchers. So bear with me. Just one other question is about your advice for young scientists and engineers in terms of choices they make. Was a mentor key in shaping your career? You did mention Judah Folkman, and also which advice of your mentor was most important? So these are some questions from some of the younger people.
Yeah. Well, having a mentor like Judah Folkman was great. And, it wasn't advice per se that I got from him, but what it was him as a role model, because he was criticized a lot. A lot of scientists said he was wrong. His ideas didn't work, and he never gave up. And I'd say the two things that I've watched that made him great, and this is what I advise people in any area, is to dream big and never give up. Or almost never give up. So those were two things that I saw, and that kind of advice is what I would give. I think there was another question embedded there, though.
Yeah, the question was actually specific to the advice of course, about the choices that they make in terms of the problems they pick or the paths they take. I think you also gave your own example of choosing to continue with healthcare when the whole world was looking elsewhere as well, right?
I think that, yeah. So I think choices, you should just do. You should never do something for money. You should do something because you believe in it, because you love it. I mean, that's what I think. I think I'm a huge believer in the happiness rule, in passion, and it could be anything. Different people like different things, and that I think has helped make science better. Some people like very basic curiosity-driven work. Other people want to get products out the door. Some people want to do both. I think it's a healthy combination, but people should, I think, follow whatever their passion is.
I think I'll just one last question and we'll stop. I realize we're out of time. Do you have any suggestions and recommendations for academic entrepreneurs who wish to set up startups? How do you manage your time and priorities? And is there a role? There's also a question on, is there a role for incubators playing a role in nurturing science-based innovation? And that's, by the way, is not from me, that's from somebody else. That it's also MIT has set up the engine and so on. Does it play a role?
Yeah. So going backwards, I think that I think incubators can be helpful. Sure. I think, you know, both in terms of funding, advice, and maybe even creating a community that people can get feedback from. I think that just trying to go remember all the questions again.
The other question was specific to academic entrepreneurs, the people who are from academic backgrounds, and how do you manage your time, priorities?
Yeah. So, I mean, I maybe do one company a year. I mean, that's how I probably would look at it. But I'm also working with my students or former students. And that really makes it easier for me because they're, you know, I probably would be talking with them anyhow. I've really wanted to be help on their careers. So, I mean, I work real hard. But also I would say that the most time that a company needs you is in the beginning. I often think of companies as like, if any of you are married and have children, it's like it's having a child, like right when it's a baby, they need a lot of your time and a lot of your nurturing. When they're 2 or 3 years old, they need less. When they're 10 or 12, they need less. When they're sort of grown up, they may not even want your time or want you to help, or they might. And actually, to me, it doesn't matter. I just want a happy child or a happy company. But, you know, usually the time that you spend the most on is in the very beginning.
I think, Professor Langer, we have run out of time. So thank you so much. We really appreciate it. I've come to the end of the planned session, and I wish to sincerely thank you for taking the time out of your busy schedule to speak on this occasion. Sorry, we couldn't take up individual questions in the interest of time, but I hope we have covered some broad topics, at least of interest to students and young researchers. I think your life's work is an inspiration for all of us, and we hope there will be many researchers, innovators, entrepreneurs in the audience here who will draw upon the inspiration to build their own impactful solutions and enterprises addressing the most pressing problems of the world. So I think with that, we'll stop here. Thank you very much. Thanks a lot.
Thank you all. Thanks. A pleasure. Thank you.
Thank you, Professor Langer. Thanks a lot.